dealing with COVID-19 right now, oncology, endocrinology, transplantation, neurologic disease, autoimmune, and allergic disorders. He successfully led development, operations, and medical marketing for innovative and important new product developments and product launches. He has managed successful groups at Johnson & Johnson, MedImmune Inc., and Auxilium Pharmaceuticals. He has also acted as the CEO and a board member at Calibrant Biosystems Inc., and more recently as Chief Scientific Officer at Green Fence Holdings, a company focused on cannabis development. For the past 20 years, Bob has been managing Hirsch Biopharma and Hubsco Consulting LLC, during which time he has worked with venture capital groups, technology transfer offices, and a number of small businesses. In addition to all that, he now is spending time developing novels and other books that draw on his accumulated life experience. His first novel, which he may give you a teaser of tonight, but which he's discussing at an NJC Connects event on October 15th at 2 p.m., is called Invisible Threat, a bioterrorism thriller that was recently published by iUniverse. Bob received his AB from Brandeis University in Boston, his doctorate from Georgetown University, and he continued his academic career at the Johns Hopkins University School of Medicine and the University of Maryland School of Medicine before venturing into the pharmaceutical industry. And now I'd like to present Bob Hirsch talking about what they don't tell us about viruses.
Okay. Thank you, Stephen. I hope I can live up to that. That took a long time. Wow. I'll have to cut that down. All right. Well, this is one of the titles that was thrown out there. There are a lot of titles we saw to this presentation during the last few weeks. The truth or Emmett about viruses and the immune system. I'm not sure there's any truth in biological sciences because every day we see something else coming out. Things are moving so fast that every night I'm reading and Amy says, "Can't you put that down? We're watching something." And I'm reading, and I asked her what happened on that Netflix series. I said, you're two episodes behind, but I just can't. I thought I was retired from the virology business, but I can't. I mean, I've been working on this for so long, and COVID is such actually, let's get the nomenclature straight right now. COVID-19 is the disease. SARS-CoV-2 is the virus. People sort of mix these things up. Hey, you're going too fast. Go back.
I clicked it too many times for you. I'm sorry. Bob asked me to do this for him, so you're at my mercy.
Don't look. Go back.
I don't know how to go back.
Oh, geez. You're taking away all my, all right, stop right there. All right, well, anyway, science is moving so fast that what I was going to say is you can't handle the truth. Oh, here we go. And you've heard that many times before. And come on, I was going to play that. All right, you've heard that before, right? From our buddy now I can't remember the movie. Come on, help me out. What was the movie?
A Few Good Men, Jack Nicholson.
Okay, right, all right. And the truth is always changing. And I don't know if anybody knows the truth except Hashem, because it's changing all the time. All right, let's go to the next slide now. Now, I'm not sure the Jews will be blamed for this, but the Jews in the past, this is an example of the plague, the Black Plague in the 1300s. Now, we're talking about a virus here. This is a bacteria, Yersinia pestis, which killed 95 million people in the 1300s. But fewer Jews died. And I'm not sure many of you know about this, but because Jewish law prescribes washing regularly and quickly burying the dead, which were full of the bacteria, many fewer Jews died. So Jews were blamed in large part in many countries for the plague. So it prevented many Jews from dying. I don't think we'll be blamed for. You coming in here, Amy? Oh, you want to come in here? You don't see the slides? Okay, you could come in here. So Jews were persecuted. Next slide, please, Dick. So let's turn to the topic at hand, and that's what's a virus? And I'm trying to basically cover 4 lectures in medical immunology and microbiology that medical students take in about a half an hour, but we can do that. Basically, a virus is a bag of code. It's not computer code, which is ones and zeros, but viruses contain either RNA or DNA, which are nucleosides or nucleotides. There are 4 different molecules. Viruses have no ability to live on their own. They cannot live on their own. They just die. They don't live at all. They're just pieces of these nucleotides, either wrapped around, and I'll show you some pictures, wrapped with lipids and membrane, which it gets from the host, any animal cell, or protein. They're really a parasite in the real sense of the word. And without another organism, they cannot exist. However, if they're too successful, they can't exist either. If it gets into an organism and kills it off, it can't get to the next organism. So that virus is not very successful. Think of a virus that kills off an organism, but can't get to the next organism. Okay, so next slide, please, Dick. This is what you see all the time as the the SARS-CoV-2 novel coronavirus. The inside is the inside blue area is the nucleocapsid, and the things that are sticking out are the glycoproteins that stick to the host cell and bind to the host cell and then get into the cell. These are the targets that we're going after with the vaccine, that we can block block entry into the host cell. Next slide, please. This is what it really looks like. And it's called the coronavirus because it looks like a crown around. This is a virus magnified 100,000 times by a transmission electron micrograph. Just want to show you what it really looks like. Next slide. So those spike proteins on the surface, those are green, right, Amy? I'm colorblind. That's green sticking out? Okay. Those are the spike glycoproteins. That's, for the most part, what the vaccine companies are going after. Something that can bind to that and block the binding to the host cell. Inside the squiggly things, that's pink, I believe, that's the RNA. That's the meat of the virus that gets into the cell, takes over the cell machinery, and replicates. It replicates, and once one virus gets into the cell, that one virus will make 100 to 1,000 viruses in one cell and spit it out and go to other cells and replicate and replicate and replicate. And then you have another cell surface protein, and then you have the envelope that says envelope. That's actually all lipid, and it comes from the host cell. Next slide. This is a general picture of what a virus does when it hits a cell. First, it attaches to a cell, specifically to a cell protein or glycoprotein. Then it penetrates. And once it penetrates the cell, it uncoats. And then the nuclear material, whether it be DNA or RNA, there are hundreds of different viruses. I've worked in my time with measles, with mumps, with poliovirus, with HIV, with a number of different viruses. They all have different types of nuclear material. The nuclear material goes out, and then it uses the cell machinery to replicate. And then it uses the cell machinery then to develop its own protein that it needs, and then it comes out of the cell. In most cases, the cell dies, and the cell then will spit out hundreds of viruses. So it's a real parasite, it kills off the cell. In other cases, the cell is persistently infected, and the cell doesn't die. For instance, herpes. The virus stays in there in the genome. It spits out some virus. The virus goes to another cell, and it stays in the body and stays there forever. So it doesn't kill the cell, but it stays there for a long time. And it may come out and go into mucosal tissue and affect somebody else. HIV goes into lymphoid tissue, stays in the lymphoid tissue, the lymphocytes, and is persistent infection. But it can infect other people. Next slide, please. Why is COVID different? Well, it's not that different. It's new, and because of that, it can transfer to other people quickly and replicate because people don't have pre-existing immunity. So I think that's what we want to discuss for the most part today. I should have made this title, why is SARS-CoV-2 different, not why is COVID-19 different? Again, the media is mixing up COVID-19 and SARS-CoV-2 all the time too. Again, COVID-19 is the disease. SARS-CoV-2 is the virus. Again, without any prior exposure, rapid spread ensues. And that's why we need to get people vaccinated. And I'll talk about why people who have been exposed already and have had an infection. Let me see if this, I'll turn the light on. I don't know if you can see me anyway, honestly. And we'll talk about will a vaccine help? I'll tell you right up front, a vaccine will help, but it's going to take some time. It's going to take some time to develop that vaccine. It's not going to be by election day, and it's not going to be by the first of the year. That's my own opinion. So how did all this start? Next slide, please. How did this virus start? No one knows. It probably came from a bat, a pangolin, or a civet. Now, a little joke here. If the Chinese were kosher, we probably wouldn't have had this happen because these creatures are kosher to eat. I looked that up. None of these are kosher, but unfortunately they are. I think it came by eating some of these animals that were infected. But how do they get infected? I don't think it's a coincidence that the Chinese Institute of Virology in Wuhan is right there near these markets. But was it purposeful? Was it accidental? Was it sailed by an employee of an experimental animal in the market? Those are some of the possibilities, I think. I actually think it's the third possibility. I don't know how much these employees make, but I think this is the third possibility. That's just my own thought, that someone who worked there took one of the animals they're working with that was infected with this virus and decided to sell it in the market. That's just opinion, that's an editorial. Next slide, please. How do we respond to viruses? This is coming into the immunology. We have a lot of defense mechanisms. So let's go right to the immunology here. Next slide. All right, we have innate mechanisms. Those are things, including your skin. Something can get in right on the skin. The dead skin, nothing can get into your skin. That's not even listed there. But innate mechanisms are not very specific. They're fast. Your phagocytes, the macrophages that are circulating in your blood, that are in your mouth, interferons that are produced by your leukocytes, your blood cells. They are fast. They respond. They are antiviral, but they're not very specific. They don't do a great job. And there are some things called natural killer cells, which I've investigated in the past. They're very quick responders. They respond within 1 to 2 days. They don't do a great job. They're innate responders. You also have enzymes in your saliva and your nasal passages. Again, they may knock down some viruses that are there, but they don't do a great job. Then you have acquired responses that you develop after you get hit with a pathogen. So these are much slower, take 7, 10, 14 days to develop. You have early antibody responses that they work within 7 days. Then you have T cell responses, and we'll talk about those as well. Your lymphocytes that develop and will kill off infected cells, virus-infected cells. But those take longer, and maybe by then, it may be too late. So, which is why it would be nice to have a vaccine in place to get people geared up and have those responses already in place if the virus circulates again. Next slide, please, Dick. This is some of our immune system, the anatomy of it. There are actually some things missing on here. Most importantly, the bone marrow should be here. I picked up a bad slide. Bone marrow is where your B lymphocytes, there are 2 types of lymphocytes. Your B lymphocytes, which are coming out of the bone marrow, which make antibody. And your T lymphocytes, which come out of your thymus, which is right about here. And as you age, after about 50 to 60, it's very small and involuted, but it's a very important part of your immune system. Your T lymphocytes are important in fighting virus, and they're developed early in age, and they circulate, and you still have them, and they've developed, they're still in your body. So the spleen is important. For the lymphocytes are sitting in there. Your lymph nodes are all through the body. They're key way stations for the body, for your lymphocytes. And when something is, when a pathogen is in your system, there are signals that are sent out to the lymph nodes, and the lymphocytes are called to that area, and they sort of fly over there and try to fight off the invader. I mentioned the skin as an important area as a barrier for invading pathogens. The stomach and intestines have a whole lymphoid system called GALT, the gut-associated lymphoid tissue. So there's lymphoid tissue all through your intestines. Let me just go to the next slide and show you the breakdown of the basic breakdown. I'm not sure this is showing up very clearly, but there's what we call humoral immunity, which is circulating in your blood, the antibody, and cellular immunity. And these little Y's are the antibodies. And that's what the vaccine, for the most part, is trying to induce. B lymphocytes develop from the bone marrow, and when they encounter, in the presence of other cells of the immune system, when they encounter an antigen or a virus or a bacteria, when they encounter them in the presence of T cells and other things like macrophages, they develop antibody, and they spit out a lot of antibody, and the antibody will neutralize the virus. And that's why you see the little Y's here binding these green or yellow things there, these extracellular microbes, whatever they are. In this case, it's a bacteria. And they will neutralize it, so it prevents the bacteria from getting into the host cell. So that's what the humoral immunity is all about. It's developing antibody. And it takes a lot of interactions of immune cells to do that. The cellular immunity are the T cells. These are big players. T cells they basically mature when they see an invader. They see an invader like a virus, or they could see a malignant cell. They mature and they kill off that infected cell, that virus infected cell, or that malignant cell, and they actually lyse it, they break it apart. So those are the two arms of the basic arms of the immune system, the antibody and the T cells. Okay, next slide, please. There's going to be a quiz after this. I hope you're taking notes. Now, this is something that probably until the last couple of days you haven't heard much about. And I've been talking about this to all my friends and to Amy and saying, I've been very upset about immune memory because no one's talked about it. Primary and secondary immune responses. Here's what happens. Look at antigen A. Let's call it a virus A. It gets into the system and you get a small little response. That's fine. You get a response. Let's say it's COVID. You get a response. You recover. And then antigen A comes in again and you get this huge response. And you basically don't even see an infection. You recover immediately because the body has been primed. The body's been primed, and you have something in there called immunological memory. And no one's talked about this in the media. And I would tell everybody that if you have been infected with an agent, you're not going to get reinfected. If you're recovered, you're not going to get reinfected because you have something in your immune system called memory. It's better than your brain. You don't have to think about it. Once your immune system sees something, it's going to remember it. Next slide, please. All right, let me tell you a little story about a carpenter, measles virus, and immune memory. There was a carpenter in Denmark who was sent on a job to the Faroe Islands. He had a cough and he was sneezing. And he went to the Faroe Islands, which was owned by Denmark. They're part of the Danish Empire in 1845. The Faroe Islands had 7,500 people. Lo and behold, when he got there, he had full-blown measles. It's starting to rain here. I hope we don't lose our power because anyway, within a matter of weeks, 66,000 of the 7,900 people on the Faroe Islands got measles. Okay, everybody over 60 years of age didn't get measles. That's because measles had been there 60 years ago. And those people had seen measles 60 years previously. He was the first, and Dr. Peter Panum from Denmark came and investigated that. He was the first one to describe immunological memory and the first one to do a real epidemiological study in 1845. So we've known about immunological memory and viruses for so long, and no one's brought this up. I'm convinced that I don't understand why no one's talked about this. And I will tell you that, I don't know if you've heard this story last week, but there was a ship that left Seattle, a fishing ship, a couple weeks ago. They measured everybody, they checked everybody for antibody to COVID, 120 sailors. 3 guys had antibody. When they came back last week, I think 30 or 40 guys had COVID. The 3 guys who had antibody, no, I think everybody had COVID. The 3 guys who didn't were the guys who had antibody beforehand. So this goes to show you that it works for COVID as well. Anyway, next slide, Dick. A little thing about immune senescence and aging. When you're young, you've got a robust immune system, high vaccination efficiency, resistance to infections. As we get older, we have less resistance, more inflammation, and autoimmune activation. And this is probably why we're getting higher doses of flu vaccine, the high dose flu vaccine as we get older. So we get a little boost. So we get immune senescence. Next slide, I want to show you sort of a real, this is a response to tetanus toxoid in different age groups. I guess 60 is right about here where I'm covered up. I'm not sure that everybody can see this, but where Steve's name is where the, on my screen is where the greater than 60 is. But as you get older, your response to tetanus goes down. This is antibody response. Next slide. So I also want to tell you that the immune response is very helpful, but it's also a double-edged sword. And we've heard this in the COVID situation. We've heard about the inflammation that people are having and problems with COVID in the lungs and in cardiac inflammation. So I just want to show you the next slide. This is a picture of, because obviously it's a positive. This is where we see lymphocytes binding to normal cells when they're in an area, and you get a cytokine storm. Cytokines are released by lymphocytes, the interleukins, interleukin 2, interleukin 4, interleukin 6, the interferons, and they bring in more lymphocytes. And what you have is a huge inflammatory response, which are causing the myocarditis and the lung issues that we're hearing about with COVID. And that's why the steroid treatments are working and are a good treatment for these late stage diseases with COVID. So it's a double-edged sword. On the one hand, you need the immune system for protecting yourself from the infection, but there could be problems. And also autoimmune disease is caused by the immune system. I think we're coming to the end. Next slide. And you've heard a lot about antibodies, and monoclonal antibodies could be to the spike protein, could be something we could use as a protection until we get a vaccine. Next slide. This is something I took from the Wall Street Journal last week. And here's a slide of a human cell that's infected with SARS. And you see the protein of the virus sticking out. I'm sorry, that's not true. Here's the SARS virus sticking to the receptor of a human cell. And in this picture on the right, you see antibody given to the patient, which is binding the virus, protecting the cell so that the virus does not infect the human cell. So a lot of people are developing monoclonal antibodies. Like, if anybody has any questions about that, I can certainly answer what monoclonal antibodies are. I've worked on those. Let's talk a little bit about vaccines on the next page. Okay, we've got several vaccines in the work. I can add the Russian vaccine as well, which I'm sure everybody would jump for joy to take that. But the Oxford vaccine, Moderna, Novavax, and Pfizer, all three of those use pieces of the mRNA either attached to a virus vector or some other vector, or within some kind of membrane. membrane container, basically. And when they get into there, that RNA is coding for that spike protein. And what you're doing is you're hoping that the immune system responds to that spike protein, and you develop an antibody response to that spike protein, so that when the individual then sees the active virus, the individual will then neutralize that active virus. So the spike protein, you'll have antibody to that spike protein, and the virus will not get into the cells. The Sinopharm and Sinovac have whole inactive viruses. And I actually was surprised that no one in the States were using whole inactive virus. If I were developing a virus, I might have tried that, developing a whole inactive virus. That's how some of our initial viruses, vaccines in the States were developed, with whole inactive viruses. There's probably potential for more adverse events, but you're getting all the fractions of the virus in there. These are the first four are more precise, but the Chinese are already injecting their whole population with these. The Russian vaccine is also in a adenovirus vector, which is a cold virus, and it's a I don't know too much more about it than that. So I don't think too many people know much about it. Next slide. This is basically what happens in vaccine testing. We're skipping phase 2. You do preclinical testing, basically, if you can, to see if you can put it in animals, to see if they develop an antibody response. I know many of these vaccines have been put in primates, and they've developed neutralizing antibody. They've also challenged these primates with a virus, and they've been protected. They've done phase 1 testing in humans, basically safety testing, and these humans have developed antibody, neutralizing antibody, which means you can take out the plasma or serum of these patients and then mix it with the virus in a laboratory, and shows that the virus is neutralized. So they know that it works in the laboratory. They've jumped right. Phase 1 studies have been on the order of 500 to 1,000 patients, which is more than usual for a phase 1 study. Phase 2 studies, they skip right over those, and they've gone to phase 3 on the order of 30 to 60,000 patients. Johnson & Johnson just started theirs today. It's a 60,000 patient study. I've left out some things here. Obviously, you need commercialize it. In this case, it's not much commercialization because the government has bought most of the vaccine, but production is a big issue, and they'll have to have that underway and get that all squared away. That's going to be a big issue, a huge production. Let's go to the next slide, which is an important one on developing a herd immunity. You've all heard about herd immunity, I'm certain, but this just explains it. We're all part of the herd. If no one has protection and a virus comes in, then everybody is going to be able to spread that virus to everybody else. So that's the top layer there. When no one's immunized or no one has seen the virus, and that's what the case was in this year. Basically, everybody, is that red or black? I can't tell. Okay, it's red, right? I can't say. All right. But when some of the population is immunized, and that's the green, then fewer people will get infected, but not very many. Still, a lot of people are going to be infected. But when most of the population is immunized, the spread of the disease is contained on the bottom. But what does that say for where we are now? Basically, where we are now is we're still pretty much on the top. Not many people are are immunized, or no one's immunized, and very few people have been infected and recovered. I think it's, where are we now? At 4 or 5 million in this country have. What's the number? You know? Can you unmute yourself?
Yeah, Bob, this is Steve McCloskey. It's somewhere in the 5 millions for
Right.
Americans.
Right, and if you multiply that by 10, that's about where we are. You can't. Basically, it's about 50 million who've been infected. So we got about 50 million people have been infected, because if you multiply it by 10, that's how many people have been infected. It's 5 million people have been tested, but it's probably 50 million who are been infected. That's the going hypothesis. It's still not a very big number. Okay, so let's go to the next slide, and I'll show you what this is the problem with vaccines and with anti-vaxxers. A lot of people don't want to take vaccines, and this is the problem. This bridge is only 99.997% safe. I think I'll swear. And that's been the problem with a lot of vaccines. And let me give you some data on influenza. Over the last 10 years, the average uptake on influenza vaccine is 40%. If we see the same uptake, and it's probably going to be lower, the recent poll that was taken suggested that only 38% of the population would take a COVID vaccine. Well, let's round it off to 40%. If the efficacy is 40 or 50%, which I believe the studies are powered for that, to see a 50% efficacy. We're not talking about getting anywhere near what you require for herd immunity. You require 65 to 70% to get herd immunity. So if you multiply 0.4 times 0.5, you're down to what, 20%. So you're going to have 20% of the population that is effectively immunized. So that's a big issue in my mind, and probably you'll hear that. You'll start to hear that. That's what you're not hearing, because that's the reality though with vaccination right now. We could talk about that. I think that may be the last slide. Maybe there's one more. Next slide. I think there's another slide or two. Oh, that's what I was going to say. That's what I just said. We can skip this. So COVID, I just want to mention COVID-19 is not the only enemy. The other enemy is right here. Next. Before the Donald, I had another pandemic in mind. And maybe I stopped it, maybe I didn't. 2 presidents were involved. There was a Muslim and a Jewish intermarriage. And I'm not going to say too much more. And this was the invisible enemy. Next slide. And we're going to talk more about that on October 15th. I will tell you that I'm going to donate 5 copies of this book to the temple, and you can write your check out to the temple to buy this. I'm not sure how we can, maybe we can give it to Richard and he can, you can write your check out to the temple. The list price of the soft cover is $13.99, so you can write your check out for any amount. I also put my website up there. I put up updates on COVID on my website at least every week. And I look forward to talking about my book and how I got there. It started up when I saw an uptick in measles virus infections, because I worked on measles virus for many years, and I was very upset when I saw huge increases in measles virus. And that's about all I'm going to tell you. So thank you very much, and Shabbat Shalom. Any questions?
Bob, I have a number of questions, but thank you very much for a very enlightening presentation. So let me ask a couple of my questions, and I'm sure other people have some.
Okay.
You said that we have not heard very much at all about immune memory.
I don't think they put much out about that. I haven't heard anything in the news about that.
No, I haven't either. So that's the memory your immune system creates when it encounters some kind of a virus. It
Anything, virus, bacteria, anything, when you see any foreign material, right?
Now, does that last a lifetime?
Yes, it does.
Okay, so
That was my point about the people who are over 60 who didn't get measles after this carpenter came to the Faroe Islands. Only the people who were younger than 60.
So in talking about immune memory lasting a lifetime, we've heard somewhere, either in print or on the news, that some people that have actually developed COVID-19, their immunity will only last a number of months. Some people have talked about 3 months. Is there any validity to that statement?
Well, I think what you may have heard is that certain levels of antibody may have been measured and may have been decreased. That's not the same as having immune memory.
Okay. Well,
because I believe you may have lower levels of antibody for a certain time, but as soon as you encounter that pathogen again, those cells will be up and running and ticking and take off and take care of that pathogen.
Okay. And just one more question. Operation Warp Speed, which is the governmental effort, the federal government effort to develop a vaccine and get it to all of us. Do you have any concerns about the rapidity of the effort and whether it will compromise getting a safe and effective vaccine?
I don't think it would compromise it. I think the FDA will review the data as it would review any application.
Okay, thank you.
Now, whether or not these will work is a whole other kettle of fish.
Okay, thank you.
Okay.
Bob,
Yeah.
If you're talking about the fact that once you've been exposed to the virus or any virus, you have this automatic immunity, why are they saying that you'll probably need 2 vaccinations?
That just may be the way they've evaluated that way they're doing their testing. They think that this vaccine will require 2 injections, just like the herpes zoster vaccine was tested, and they think they need 2 to get the right level of immunity.
Right.
and with shingles as well, right?
That's what I heard.
Oh, it's herpes, yeah, right.
Yeah.
It's not the same. A vaccine is not the same as getting an actual infection. You don't get the same level. You don't get the same level of immunity.
Okay.
I have a question, and that is regarding people in our age group with the lower number of T cells being produced. What can we do to boost our T cell production? Is there anything we can do?
Well, I'm not saying we have lower numbers of T cells. I'm just saying they're, we still have them. I'm just saying they're not necessarily not as active. I don't know if there's anything you can do about that.
Bob, I have another question. It seems that testing, the issue of testing, is always in the news. Number one, how important is the issue of testing, or the matter of testing and combating or getting COVID-19 under control? That's number one. Number two, can we ramp up testing so that we can do effective contact tracing?
Oh, important questions. Very important questions. I don't know if I can answer those questions. I really don't.
Okay,
Obviously, testing is very important. I've seen it slide down over the last, few weeks, and I'm not sure why that's happened. And I'm not sure that the tests, I don't know if the tests have been accurate either recently. I've heard, I haven't been following it that closely, so I don't want to answer those questions.
okay.
We should speak.
Bob,
Yes.
there's been publicity recently about the use of plasma from infected patients with antibodies, and apparently the FDA withdrew approval. Could you comment on that, please?
Yeah, I'm not sure why. I'm a real proponent of that. In fact, back in March and February, I sent out notes to a lot of people I've worked with in the past to get your butt moving on this, because I actually worked on 2 products that used plasma-derived products to treat viral infections. So I don't understand that at all. I really don't. We developed 2 products that purified the immunoglobulin to treat respiratory syncytial virus and cytomegalovirus. So I really have no idea why they did that. No clue.
Okay.
Unless they're just using plasma and not purifying the immunoglobulin, which is where the antibody is. I mean, just transferring plasma is one issue. Maybe they need to purify the immunoglobulin, which is where the antibody is. That would be a better thing to do. There are companies that are working on that, but I don't understand that. I really don't.
Thank you.
Steve, I have a question. Hello.
Go ahead.
Here, Bob, there's a lot of articles about the vast majority of people that will not take a vaccine, and I think you alluded to that.
Right.
Is there any downside to the 40% or 50% of the people that do take it? They're still going to be protected, assuming the vaccine is 60-70% effective, correct?
I don't think there'll be a downside. We'll have to see the data. We'll have to see what the adverse events are in the clinical trials. We'll have to see what it looks like.
And it's also it is my understanding that there's at least one company in the paper that that is working on a one-shot vaccine.
Johnson & Johnson.
Johnson.
Yeah.
Johnson, a company I'm sure you're familiar with.
Yes, I'm familiar with them.
So are we.
So are we. All right, thank you.
Right. Anybody else?
Anybody else have any questions of Bob? He's a valuable resource, so ask your questions if you have them.
I have a question, Bob. Would you volunteer for one of these clinical trials?
Would I?
Yeah.
I guess I would. Yeah. I would.
Okay.
I don't know if it'd take me. I don't know what the age is. I think the age is from 0 to up to 65. I don't think they're doing older than that.
Oh.
I don't know what the age ranges are on the trials. Oh, I just gave away my age. I don't know what the age ranges are. I don't know if they're doing children either.
Okay. Anybody else have any questions of Bob?
Whoops, I just lost what happened. Am I still there?
Yeah, you're still here.
All right, I
You're
don't see my,
Anybody else have any questions of Bob? Okay, if
Wait, one more,
not-
one more.
Okay,
Yeah.
one more.
All right, one more. Bob, this virus seems to be causing many post-infection problems for patients, and children are being infected and teenagers, many things that haven't been seen or predicted before. Is there anything that anyone knows that's particular about this SARS virus that is causing these reactions?
a lot of viruses cause post-infectious problems. Influenza causes a lot of post-infectious problems. Post-infectious myelitis, post-infectious carditis. So it's not unusual. As for children, I just did a lot of research, literature research, because I did a presentation, and I did a blog yesterday on on children from 5 to 19 for schools. It's interesting that children 5 to 10 do much better than children from 10 to 19 in terms of both in getting infected and passing the virus. They're much better off in terms of the elementary school kids than the kids who are high school. I'm not talking about college age kids. But in terms of post-infectious diseases, post-infectious syndromes, that's quite common.
Okay.
Thank you.
I have a question as far as the flu vaccine
Yes.
they're saying, make sure you get it. Is that just so we don't have a second pandemic on top of the one we've got?
Yeah, I'm not sure what the flu vaccine is this year, what the different serotypes are. But yes, I would get it. Probably get it in September, October. Get it early. There's definitely going to be a problem with these two viruses circulating.
Yeah.
Have to get that.
Fran has a question.
Yeah, could you comment on any long-term after effects of the COVID virus on the body?
I really don't know. We don't know what that is.
Okay.
I wish we did, but we don't know.
What about college kids? Do you
Oh,
think
you can't control them. I would think by October 15th, they'll all be home.
Yeah.
That's right.
I think
I
I
agree.
you think it's going to take that long, huh?
Well, I
It
mean,
depends on the size of the school and how good the administration is.
Home, if you think.
yeah, because they're gonna fail. They're gonna fail to control it.
Yeah.
Absolutely.
Well, March isn't even open.
That's right.
I mean,
Anybody?
if everybody wore masks, if everybody social distance, if everybody didn't touch their face, everybody walked in a line and stayed. You can't do that. These kids aren't going to stay away and not party. That's college.
Mhm. It's true. So does anybody else have any other questions? Bob, I want to thank you very much for doing this today. It was very, very interesting, very enlightening. And we will look forward to October 15th at 2 o'clock, Invisible Threat. And I will just tell everybody that Shelley and I read the book. We found it very intriguing. We found it prescient. And it's something that you should read.
Okay, and Dick, I'll bring over 5 books to you and you can figure out how to determine how to get a contribution from people for the temple. How's
Okay,
that?
thanks, Bob. Thanks for your presentation too. It was great.
Thank you, Bob. That was excellent.
All
Very
right.
good.
Take
Is
care.
that his book, King is over there?
Bye, everyone.
Bye.
Shabbat shalom.
Bye. Thank
Shabbat
you.
shalom.